Okay, so this is chapter 15, part 3. We're still talking about transcription and translation, gene expression. And in this part of the chapter, we're going to focus on transcription in eukaryotic organisms. And so we talked about transcription in prokaryotes. Let me get to the next slide. So fundamentally, it's the same process in eukaryotic organisms like us. It's still initiation, elongation, and termination. There's just a few key differences. The first one that I'll point out is RNA polymerase. So RNA polymerase, remember, is the enzyme that's responsible for reading the template strand of DNA and synthesizing the RNA, whether it's mRNA or tRNA or rRNA. Okay. Now, in prokaryotic organisms, like bacteria, there's just one type of RNA polymerase. But in eukaryotic organisms, RNA polymerase, it's more complex. And there's actually 3 different kinds of RNA polymerase for the 3 different types of RNA molecules that are synthesized. So that's a key difference. Secondly, eukaryotic organisms, again, like our cells, they're going to have membrane-bound nuclei. And that's where transcription takes place, inside the nucleus. And protein synthesis happens in the cytoplasm. Okay, so transcription in the nucleus, but translation in the cytoplasm. And so therefore, the RNA, whether it's mRNA or tRNA or rRNA, has to get out of the nucleus to be translated. And that process in and of itself is a little bit difficult. And then once it's in the cytoplasm, it's got to be protected from degradation. So there will be other enzymes inside the cytoplasm that are going to be just waiting to kind of chew up RNA. And so it has to be protected from that process. And so we'll talk about what happens to our RNA. Okay, so first off, let's look at some major differences in initiation. So initiation is, fundamentally the same process, right? You have RNA polymerase, it's going to bind to the DNA, it's going to create a transcription bubble, and then it's going to get started. But again, it's a little bit more complex when we're looking at eukaryotic organisms. So first of all, in eukaryotic organisms, it's more than just sigma factor. Remember, sigma factor was there for prokaryotes. In eukaryotes, there's a bunch of what we call transcription factors that bind to the promoter region and recruit RNA polymerase, the correct one, to that spot. Okay, so remember, the promoter region is going to be a region upstream from the transcription start site. There will still be a conserved sequence that I'll talk about in just a second, but that promoter region, so if I look at the picture here, here's my promoter region right here, starting here with this green and arrow proceeding this way. This is the gene. Okay, so our gene is here and it will be right in this direction. So upstream of that gene, so before the gene, is the promoter sequence. Okay, and actually let me mention the conserved region here. So in eukaryotic organisms, at about the -30 site, there is what is called the TATA box. This is a conserved sequence. So again, for all eukaryotic genes, about 30 nucleotides up from the start of the gene will be this conserved sequence. Okay, so similar to what we saw in prokaryotic organisms, but not the same sequences. So what happens at this promoter region? Again, transcription factors. So TF2D, TBT, TF2A, TF2B, TF2E, TF2H. Okay, all of these transcription factors will bind, which draws in the correct RNA polymerase. So in this case, it's RNA polymerase 2, which is the RNA polymerase that synthesizes mRNA. Okay, so same concept. You have a conserved region in the promoter, which is going to attract these proteins, these transcription factors, which will then attract RNA polymerase. Okay, so again, basically the same with some subtle differences. Elongation, again, is essentially the same as it is in prokaryotes. So again, with mRNA, we have RNA polymerase 2. Okay, it's going to, again, synthesize in the 5' to 3' direction, just like we saw in prokaryotes. However, the RNA that starts to come off and starts to be made, we're going to call it pre-mRNA because there's going to be some changes that are done to that pre-mRNA before it's actually going to be called mRNA, before it can actually be used. Okay, so we'll talk about those in just a second. I also just want to point out, because there are histone proteins, so remember, eukaryotic DNA has protein associated with it. Our DNA is actually wound around these proteins called histone proteins. Those proteins are there to keep our DNA, organized. And so during elongation, our histone proteins have to be moved in order to access our genes. Okay, just like when we talked about replication, we've got histone proteins to contend with. So the process is just a little bit more complex in eukaryotic organisms. Termination is different depending on the RNA polymerase, but there's going to be, at the end of the gene, what we call a termination signal sequence, or just sequence. And that's going to trigger termination. So I want to quickly talk more about this pre-mRNA and how it is processed. Okay, so again, pre-mRNA, it's got to be processed a little bit before it can be translated. And the same goes for tRNA and rRNA. Before they can be transported to the cytoplasm, there has to be some things done to the molecule first. So there are three things that are done to the pre-mRNA, or pre-tRNA, or pre-rRNA, whatever type of RNA we're talking about. The first is the addition of something that we call the 5' cap. So at the 5' end of our new mRNA transcript, a cap goes on it. What's that cap made of? It's made of 7-methylguanosine. Okay, so 7-methylguanosine. And I'm going to show you a picture that has this in it. Why? Why does this 5' cap get added to our transcript? So this actually protects the transcript from being broken down in the cytoplasm. So that's what that term degradation means. It also aids in its recognition by the translation machinery out in the cytoplasm. So what is that? That's the ribosome. Okay, so this cap helps the ribosome to bind properly to our transcript. Okay. Secondly, at the other end of the mRNA molecule, the 3' end, we add something called the poly-A tail. And so there's this enzyme called poly-A polymerase, and it is going to add a string of about 200 adenine nucleotides to the end, the 3' end of our transcript. Okay, and not part of the gene. These are just added parts that will not get translated later. Again, why? So again, to decrease the chances of this being broken down or degraded in the cytoplasm. And secondly, it's believed that this poly-A tail, this 3' poly-A tail, also helps the transcript to exit the nucleus. Okay, so it helps it get through the nuclear pore complex and into the cytoplasm. So those are the first two things. There's a 5' cap and a 3' tail. Lastly, I'll show you this here. Well, actually, before I show you this next part, let me just show you. This is the pre-mRNA. And so at the 5' end, we put this cap, this 7-methylguanosine cap. And then at the 3' end, we add this poly-A tail. So those are the first two modifications. The last modification is a little bit more complex. And so this is only true of eukaryotes, all of this. So, these three things are only true of eukaryotes. But in eukaryotic genes, there are regions called exons, and there are regions called introns. Exons are coding regions. They're actually part of the gene that needs to get translated. Okay? Sorry, my dog's barking. The introns will interrupt these exons. And the introns are not part of the coding region. We used to say that introns were junk. We weren't sure why they were there. Through evolution, there's been replication of the genome, and these pieces just ended up there, and we're not sure why or what they do. There's more recent evidence that says that that's probably not true, and that these introns are really probably involved in gene regulation. They do have a function. They're not just junk DNA. And so again, there's more research coming out about that. What we do know for sure, though, is that the introns get removed and the exons get spliced together during this pre-mRNA processing. Okay, so we get a 5' cap, a poly A tail, and we take out introns. Okay, so that's shown here again in this figure. So the introns are shown in the silvery gray color, where the exons, or the coding region, are shown in blue. So we got one exon here, a second exon here, and a third exon here. And so the introns get taken out, and we end up with our ready to be translated mRNA. This can now exit the nucleus, go to the cytoplasm, and get read by the ribosome. So again, it has a 5' cap, a poly A tail, and here is the message that is just made of exons. Okay, so that concludes this part of the chapter.